Hello to anyone who's watching this. Thank you for taking time out of your day to listen to my research that I've been able to do. We will be going over the effects of pharmacological inhibition of AMPA receptors in the nucleus accumbens and the basolateral amygdala on the reinforcing properties of alcohol in male C57BL/6J mice. So we're going to set a little bit of a scenario to get some background knowledge before we dive into the research. So we're going to start with a little girl named Sally, and Sally is at a party or whatever with her friend Bob. Bob offers her drink, and she takes the drink and she starts drinking. And over the next long period of time, we notice that she starts drinking over and over and over and over again. And until she has really no control over her alcohol use and has a negative emotional state when she's not using it. She, like the other 16 million Americans in America, suffer from AUD, or alcohol use disorder, which is a chronic relapsing brain disease characterized by compulsive alcohol use, loss of control over alcohol intake, and a negative emotional state when not using. Unfortunately, due to ineffective treatments for AUD, she dies, and she is not the only one. Worldwide, 2.5 million people die annually due to harmful alcohol use. So we want to know kind of why did Sally keep repeating the behavior of drinking as casual alcohol use, and how did it progress to uncontrollable abuse? So let's look inside her brain. So this is Sally. This is this is her brain before alcohol use. There's GABA, which is an inhibitory pathway, and then glutamate, which represents the excitatory. When she drinks alcohol, there is more GABA, and this creates an imbalance in the brain. After drinking chronically for long periods of time, the brain will try to fix this balance by increasing the activity of glutamate, and that basically balances out the inhibition with excitatory. But when she stops drinking, GABA goes down because you're not drinking alcohol, but glutamate stays up and is super hyperactive. And is actually this overactivity of glutamate is one of the reasons why we see seizures as a withdrawal symptom, just due to overexcitement of specific brain areas. And a reason why we care about the overactivity of glutamate is it is a contributor to the alcohol dependence and continuation of alcohol seeking behavior. So what research has been done on alcohol or glutamate in relation to alcohol? So this is glutamate and the all different types of receptors that produce glutamate. So currently there's been good research on the AMPAR receptors, which stands for alpha amino 3 hydroxy 5 methyl 4 isopropionic receptors. So currently we can see in research that AMPARs play a role in the regulation or reinforcement of other drugs of abuse. And these drugs have been, just to mention a couple, is cocaine and heroin. And so as you can see from the figure that AMPARs are made up of 4 subunits, and what's interesting about these receptors is not every AMPAR has all 4 of these subunits. They can be a mixture, and that affects the activity of those AMPARs. So we are interested, we see AMPARs play a role in reinforcement of other drugs of abuse. So what about alcohol? So far what we've seen for AMPA research has been focused on the nucleus accumbens. This is because it's a main reward center in the brain, which means for this reason, there is a lot of, it's a convergence site for many glutamatergic projections. As you can see in this little graph, there are just 2 regions, it's simplified, but you can see the glutamate is going to the NAC nucleus accumbens. So what we've seen so far with that is alcohol increases the expression of the GLUA2 subunit. So this kind of makes us think, what about makes us want to focus on GLUA2 containing AMPARs in this region. And one of those where the glutamate comes from is the BLA, as seen on this little figure. BLA stands for basolateral amygdala, and as you can tell, it sends some of those glutamatergic projections to the NAC, and we have seen that alcohol increases the expression of the GLUA1 subunit in this brain. So that makes us want to look at the GLUA1 containing AMPARs in the BLA. So this is like a little depiction of those two and which regions we are interested in. So our objective for this study was to determine if the inhibition of GLUA2 containing AMPARs in the NAC or the GLUA1 containing AMPARs in the BLA regulate the reinforcing effects of alcohol. And so let's talk a little bit about what I mean by reinforcing. So reinforcement is a concept or is a fundamental behavior process that is the frequency or probability of a response is increased by a dependent relationship with a stimulant or circumstance. So basically what this means is the frequency of a behavior will increase the response and if this stimulus is seen as rewarding to or a pleasurable experience for the subject. So we would see a repeat behavior to obtain the stimulus. That means it would have some reinforcing effects. This is seen in all species. So that means we are able to use animal models to understand more about reinforcement in humans. And specifically for alcohol reinforcement, we use an operant self-administration mouse model. So what that means is we take a mouse and we teach it in some training to do a behavior that's not they normally don't do. So in this little scenario, we have a button. They learn that when they press the button, they receive alcohol. So when we put them in these little chambers where they're given access to the button, they repeatedly press the button. And so we can see from that the stimulus, which is alcohol, has reinforcing effects. So a reason why we use a mouse model is because, as you can see from this figure, that there are some physical similarities in brain composition of the mouse compared to us. So it makes us a good animal model for us. So this is our setup for our experiment. We started with mice, and they learned to active lever press for either ethanol or sucrose. There was, they had 40 operant sessions, and they were each 1 hour. And then after the operant sessions, all 40, they underwent stereotaxic surgery. So basically, we anesthetized them, and then we implanted little cannulas into their brains. And there was 2 types of cannulas that we used. We used ones that were aimed for the NAC, and then the other one was aimed for the BLA. And then after they recovered from that, we started microinjections. And so basically what how that worked is we started with just injecting ACSF into their brains, which is kind of like a control. It's the vehicle we use for the pharmacological compounds, and it gives them just to habituate to the process. After they were habituated to like the ACSF, artificial cerebral spinal fluid, they either had injections of PEP2M or NASPM, and as you can see, the PEP2M was directed for the NAC, and the NASPM was directed for the BLA. So after they received the microinjection, they went back into the operant chamber to active lever press for either ethanol or sucrose. And they did this a couple times at various doses, which you will see on the next slide. And then after they finished at those specific doses. We repeated the first step of doing the microinjection, but only for 1 dose. And then they were put into a locomotor chamber, which tracks their movements and can quantify how much they've moved during that 1 hour in centimeters. After locomotor sessions, we perfused the mice and then collected their brains for histology analysis. So that was a lot. So I've kind of broken down our 2 experimental groups, and the 2 experimental groups kind of have 2 subgroups within that. So that first panel that you saw, like the first top of mice, they had the cannulas directed at the NAC. We had groups of ethanol and sucrose reinforced mice within that group. There was 8 ethanol, 9 sucrose. They had the injection of PEP2M, which does the inhibition of GLUA2 containing AMPARs. In the last little column represents the 2 doses that they received, both in microgram per half microliter. And then the experimental group 2 was the BLA region. We had 6 ethanol, 7 sucrose, and they received the microinjection of NASPAM, which was the inhibition of GLUA1 containing AMPARs, and their doses were 5.6 and 10.0. So let's look at the data now. So experimental 1 group, we're gonna start with the first experiment and then move on to the second. So this first one is our ethanol reinforced mice with the 1.0 and 10.0 PEP2M into the NASPAM. We see the cumulative active lever presses throughout these 60 minutes that they were in the operant chamber. And we see that the 10.0 dosage actually decreases the active lever pressing compared to the ACSF. Then we checked their locomotor activity, and we saw that the ethanol reinforced mice, the injection of PEP2M at the high dose of 10.0 did not affect their locomotor activity, as seen in their total distance traveled in centimeters. Now we're going to look at our sucrose reinforced mice of experiment 1. And we saw that the microinjection of PEP2M into the NAC did not affect their active lever pressing at all at either dose compared to ACSF. Their locomotor activity also was not affected by 10.0 PEP2M. And now at the end of the experiment, we were able to make a stacked plate, basically just show the actual injection sites for these mice. Unfortunately, we only have the stacked plates made for the sucrose reinforced mice. We are currently still doing histological analysis for the ethanol mice. Due to the current situation we're in, we're not able to finish that right now. But this is a nice coordinate that shows that the injection site was into the NAC. So we know that the PEP2M was going into the NAC to inhibit the GLUA2 containing AMPARs, at least for the sucrose reinforced mice. So now on to experiment 2. So the first graph, we're working with our ethanol reinforced mice first, and we can see that the 5.6 and 10.0 dosage of NASPAM into the BLA significantly decreased the active lever pressing compared to ACSF. And then we looked at their locomotor activity, and NASPAM did not affect the locomotor activity of these ethanol reinforced mice. And then we're looking at our sucrose reinforced mice, and as you can tell, the 5.6 and 10.0 dose of NASPAM also decreased the active lever pressing as from the same as the ethanol reinforced mice. They both were decreased with NASPAM. And then their locomotor activity was not decreased by NASPAM into the BLA, like all the others. As for cannula placement, again, we were only able to unfortunately have our sucrose reinforced mice histologically analyzed right now. But this stack plate shows us that the injection sites were in the BLA, at least for the sucrose reinforced mice. So what does this all mean? From these findings, we suggest that the GLUA2 containing AMPARs in the NAC help regulate the reinforcing effects of alcohol. How did we get there? We saw that the PEP2M into the NAC decreased active lever pressing, and PEP2M into the NAC did not affect locomotor activity. So why we are looking at locomotor activity every time we do microinjections is we want to be able to just call out or not or deduce that it is not the alternative interpretation of these pharmacological compounds affecting the overall mobility of these mice. Because a good alternative interpretation could have been without these locomotor is, oh, these pharmacological compounds are just affecting the movement of these mice. That's why they're not active lever pressing. But these locomotor results show us that they are still moving as much as they normally would, but they are choosing not to active lever press. So back to the other results. What specifically is interesting about these GLUA2 containing AMPARs is they did not, the inhibition with the PEP2M did not affect the self-administration of sucrose. So this kind of suggests to us that these GLUA2 containing AMPARs in the NAC might be specific to alcohol. As for our experiment 2 conclusions, we suggest that GLUA1 containing AMPARs in the BLA help regulate the reinforcing effects of all rewards. NASPM into the BLA decreased active lever pressing for ethanol and sucrose and was not affected by the locomotor activity. So our future endeavors would definitely include, at first, completing the histology for the ethanol reinforced mice, just so we can get our verification of that. And then repeating the study would definitely be beneficial since we had a low sample size and we would want more confidence in our results. And then it would be very interesting to compare our results to a female cohort of mice. And if you didn't notice, we had an issue with our locomotor drug doses for NASPM. One was for 3.0 and one was for 5.6. So it would be nice to repeat and have the same dosage, so there's the similarity and not a confounding variable. And there also should be a more research into these GLUA2 containing AMPARs in the NAC, since they are the ones that were their inhibition decreased ethanol self-administration and not sucrose. So these could be more specific just to the alcohol reinforcement, but the GLUA1s, since they reduced ethanol and sucrose, those could be just more for all general rewards. And this alcohol specificity could be important because it could potentially be a drug target in the future. So acknowledgements, I would like to thank Dr. Hodge and the entire lab there, especially Dr. Fasidamo, who was my research mentor, and then my honors thesis class, Dr. Maddox, and the whole entire alcohol and behavior peer group, and the NIAAA for supporting this with the grant. Thank you for listening.